the world now is a big problem of something called as MDR-TB. It means multi-drug resistant TB, right? And also TB that combines with HIV. Now, in this combination is particularly deadly, and as you can see, India, both TB is a big problem, MDR-TB is a big problem, HIV-TB is also a big problem, right? Again, there is a particularly striking image that I will show you in the next slide. What people realized is that in the case of a bacteria like TB, if you just give one antibiotic, the bacteria will develop resistance fast. So, can we combine different antibiotics and give them? In that case, the chances of bacteria getting resistant for one of them is high, but the bacteria has to be resistant to all of them to become multi-drug resistant bacteria. So today we have antibiotic that targets different types of pathways. So we have isoniazid and ethambutol that are affecting different types of the cell wall. Then we have rifampicin that inhibits the RNA synthesis, and different mechanisms are targeted, and these antibiotics are given in combination. That's very important to understand. Now, despite of this, as I told you in the last slide, drug resistance against TB has become a very big problem. And I will skip this slide. Just take a look at this image. This is an image I took from the London Science Museum website. This, for me, is an incredible visual shock. So what this shows is a box containing drugs. Now, these are the drugs that are taken by people who are unfortunate to be infected by drug-resistant TB. So if you get drug-resistant TB, you cannot take the normal drugs, the combination of rifampicin and ethambutol and isoniazid that I told you, but you have to take a higher combination, a stronger drug that will have more side effects. You will have to take that. Now, you can see this is one drug, and you can appreciate there are many such drugs, and you can also appreciate how many drugs are there here. Now, I am not joking here. This entire volume of drug is taken by one patient during the course of his or her treatment for multi-drug resistant TB. And now this takes place over a period of 2 to 2.5 years. Now imagine the kind of difficulty the patient must be feeling to have to do. These are not pleasant drugs, okay, because they have a lot of side effects, etc., about which I'm not talking today. Now, if a patient has to take too many drugs, imagine also the pressure it will have on the system itself. But that is now a requirement. It's exactly the same as somebody having to shout to call you because you are not responding when you were called softly in the first time. Right? So this is really we shouting to kill the bacteria. But even more dangerous now is that there are new strains of TB that have come which are even resistant to this, the so-called extremely drug-resistant bacteria. XDR is called. So there's a question, if we take in combination, is there no change that they will become resistant to all the antibiotics? So there is a definition for what is multi-drug resistant. If it becomes resistant for one of them or two of the treatment, then there are different kind of gradations there. Another question, is combination antibiotic for a single infection a result of microbes become resistant to multi-drugs? This is right, as we are allowing microbes to develop resistance to drugs when we do not really need multi-drugs. This is a very good question, and I think very important question. So I took the example of TB, but TB is a bit of an unusual bacteria. It's a slow-growing bacteria, and it's a very powerful bacteria in the sense that it has infected. It's called the most successful bacteria. So for a typical cold cough kind of disease, or you will take a treatment for 1 days. But for TB, even the normal treatment is 6 to 9 months. Okay, so therefore the treatment regimen etc. are particularly carefully curated for TB. So, but then the idea of combination therapy with multiple antibiotics is also something that is actively pursued in other infections. The logic being what I exactly told, that the chances of is hopefully there should be more and more research. So a lot of antibiotics that were originally discovered in the 50s and 60s came from the environment, right? From soil, from elsewhere in the environment. Now people are exploring nature in more and more sophisticated ways. So, for example, when in deep sea floors, in pristine areas where which are in medical cure. So, can we apply now the methods of modern medicine, modern science, to understand how these cures could be working? And so that is one area people are working on, and I think there should be more research in this direction. The other area is something called as phage therapy. Phages are viruses. These are viruses that kill bacteria. Right now, if the bacteria is causing disease to us, can we not use the viruses that kill the bacteria to kill them? Right. So in that way, it's like we are clearing the bacteria. So it's like a bio mediated killing of the bacteria. So I should say this. Some particular rivers, for example, were traditionally known to have some kind of a curative properties, or water from a particular area was supposed to have some kind of a curative property. And so, doing some research, people found that these environments also harbored a lot of these phages. Now, just like the bacteria that produce antibiotics, etc., these phages are also part of the nature. And so it's part of the same language that keeps going on everywhere. Now, can we identify bacteria, or can we identify these viruses, these phages that will kill a particular type of bacteria, and can we use them for therapeutic purposes? So that's another line of thought. It's not a new thought. It's been a very old idea about phage therapy. But because antibiotics came into play in 1930s, 40s, and 50s, the work on the interest in phage biology sort of waned down, and I think that is one area that is seeing a revival now. Another possibility that is happening in my lab, particularly, is working actively on this, is something called as a host-directed therapy, where we are working with the concept that the bacteria needs our system, our body, to cause a disease. Right, so it is using our body to live and to multiply inside our bodies. Now, all these antibiotics, they kill the bacteria directly. They attack the bacteria directly and kill them. So we are thinking, instead of that, can we not find out which part of what is it in our body, in our systems, in our cellular processes that the bacteria is using, and try to not let it use that. So that's a whole new concept where we are not directly targeting the bacteria, but we tweak our human host in such a way that the bacteria cannot use us. So these are some of the ways people are thinking about how to solve, not necessarily solve, but in a sort of a post-antibiotic era, how can we encounter the future? So there's a question. Colistin is said to be the last antibiotic today. Is India working on developing new antibiotics? If so, what are the new avenues searched? Are we in a stage to get new antibiotics in the near future? So that's a good question. I think that is what I just covered. I think my direct answer for that will be that we should not be a single approach, but rather a multi-pronged approach where one or a combination of these approaches must be used. Yeah. Okay, so this is example of what I told you about phages. There was a lot of interest in phage in the beginning, but with the advent of antibiotics, the blue bar, the interest in phages had waned, but now it is making a comeback. I think I will stop here because then I would love to take some questions from the audience. There's a question: what makes the bacteria acquire its resistance faster? Is it overdosing or partial dosing? That's a good question. It cannot be overdosing because overdosing will kill the bacteria. The major culprit is underdosing, because if the bacteria is exposed to the antibiotic, it is seeing the antibiotic, but in doses that are not sufficient to kill that, right, then the chances of the bacteria acquiring resistance becomes very high. Yeah, so I think I was I hopefully I conveyed you some message about antibiotic, and I would love to hear your thoughts. Particularly, I would love to hear from students now that we are in this messy situation, what we can do to get out of it.
Thank you, sir, for your very highly informative and enlightening talk. And there are some questions by some of the participants. One is from Prakriti K. If we take combination, there is no change that they will become resistant to all the antibiotics gradually.
I think the question to the chat box I have already answered as I went along.
Okay.
This is a separate question.
Then next question is. Hello?
Yeah. Yeah. So, recent experiments prove that the dead bacterial DNA can alter the live bacterial DNA when in contact. Exactly. So, I did not go into the details of this. This is also along the lines of the horizontal transfer that I mentioned. So the antibiotic resistance genes are also everywhere in the environment. Now these genes can spread from one bacterium to the other, and it can result in a wide spread of antibiotics across the bacterial species. So resistance will develop when there is a constant exposure to antibiotic at subtherapeutic doses, and once it develops, then even dead bacteria they can spread it. What about phages? Any changes that they can also become resistant? Whether bacteria can become resistant to phages? Quite likely yes. But phages also work by different ways. So phages will inject the nucleic acid into the bacteria, and there is a whole spectrum of the natural interplay there. So there is bacteria have these restriction enzymes that can then identify the viral DNA and attack them. So there is a whole spectrum of possibilities happening there. But whether the bacteria can acquire resistance to phages, I think they can. But again, there will be so many types of phages and so many types of mechanisms in play there. And I don't think we have a complete knowledge of all the mechanisms in play. So the CRISPR system that won the Nobel Prize this year is only one of the restriction enzyme that I told you about, or the CRISPR that I just now told. They are all some of the well-known examples in nature on the interplay of these are bacterial difference pathways happening in nature. But there will be many more. we will get. So that's a fascinating area for research. So if we find out more and more such methods, I think somebody has to mute. So I was just saying that if we find out more and more about such mechanisms, we will know what are the so many ways by which we can attack the bacteria. What is your opinion about TB patient with the help of ethnopharmacology? I think what you're alluding to is the kind of you see how do drugs come from? Drugs come from a lot of drugs come from nature. So a lot of these antibiotics are coming from nature, from other microbes that produce them. Similarly, there can be other plants, plant-derived products, natural products coming from various sources, which can be screened. And whether there are any specific antibiotic or antibacterial component that is present in such extracts, it can be discovered. I think Ayurveda, etc., will have certain way of treating TB patients, but that is not very mainstream. So I think there must be detailed. See, modern medicine also has a certain criteria of how drugs are accepted. So I think we have to bring conventional, traditional medicinal approaches and modern medical research practices into some kind of a common ground.
Okay, some more questions. Now, I would like to ask one question from my side.
Sure.
In your talk, you have highlighted that antibiotics are a kind of chemicals produced by bacteria. But however, in nature, a product turmeric is also being used as a form of antibiotic. So, will there be any chance that if we use antibiotic that is turmeric for a prolonged use, will there be any chance for development of superbugs?
I don't know actually if turmeric is directly antibacterial, but what a lot of plant-based products do is also to boost our metabolism, to boost our immune system, so that we can fight the bacteria better. Right? You see, even with a bacteria like TB, and now we know this a lot with COVID. Not everybody who is infected gets the disease. Right? Why? Because we have our bodies have capacity to fight the infections, and we are doing this on a regular basis, on a daily basis, and we are not aware of it because we have successfully fought the infection. So what a lot of these plant-based products do is also to boost our own metabolism. This is in the direction of the host-directed therapy that I was mentioning.
Sir, one more question. For one particular ailment, when one particular antibiotic is prescribed, is there any chance that due to that other bacteria can they develop become as a superbugs?
Yes, they can, because that depends on the prevalence of the resistance. Now, as I told, the resistant genes can go from one bacteria to another. A different type of bacteria they can called as horizontal gene transfer. Now, these resistance genes are also ubiquitous in the environment because of our overuse of the antibiotics. Right? So, in poultry farm, for example, or in cattle, in dairy farm, everywhere, right? There's a lot of antibiotic in the environment. Now, as a consequence of this, a lot of resistant organisms are also there in the environment. Now, this resistance is a function of the genes that I told you about. Now, these genes are also therefore all over the environment, and these genes can jump from one species to another. They are called as mobile genetic elements. They can jump from one species to another. So it's entirely likely that one species of bacteria that has not been exposed to a particular antibiotic can develop resistance to that antibiotic. That is the most dangerous part.
Okay, sir. Sir, one more question. For some particular ailment, a medical practitioner A prescribes a medicine or an antibiotic for about 3 to 4 months. If it is not cured, patient goes to another medical practitioner. He will prescribe another antibiotic of his own choice. Then, with this kind of situation, will there be any chance for development of superbugs because of the different antibiotics for the similar alignment?
See, when you say different antibiotics, we have to be clear what we are talking about, because if the brand names are different, right, it is also quite likely that it is the same antibiotic, but just that the packaged and branded differently. Even if it is a totally different antibiotic, if the antibiotic is working through the same mechanism, that will be different, right? So if this is what you are asking is particularly correct, because this is exactly how antibiotic these are fertile breeding grounds for antibiotic. So if a particular antibiotic has not worked in a patient for 4 to 5 months, for example, we have to really see why, right? For which we'll have to do some kind of an investigation on that. But that is something that does not happen. And what happens is exactly what you told. Then the patient will say that, okay, this doctor is not good, he will go to another doctor. So without actually investigating the cause of why a particular antibiotic has not worked, he's moving to the next doctor. And the next doctor will probably know that one particular antibiotic did not work. And instead of figuring out why that has not worked, he might prescribe another antibiotic. So what has happened essentially is the bug inside this patient has now got exposed to that antibiotic for 4 months, right? So that is sufficient time to acquire resistance. So in all such cases, ideally a thorough investigation must happen. But how feasible it is, I don't know, unfortunately.
Sir, one more question. There is a information that coronavirus needs a ACE2 enzyme for its survival when it enters the cell. Have we identified such enzyme that bacteria seeking in the host your host directed therapy?
Oh yeah, so this is known for a lot of bacteria. So ACE2 enzyme is what we call as a receptor. That is something that host protein that the virus is using to get inside the cell. And such receptors are known for many bacteria. And in my own lab, we are not looking at that, but in some way we are looking at how we can modulate how the bacteria is coming inside the cell. So in TB bacteria, for example, can we prevent it from getting inside the cell? So what are the mechanisms by which the bacteria are getting into the cell, and how we can prevent that? That is something that we are doing research on in my lab.
Sir, one more question.
Sure.
As you said in your talk about phage therapy, will the bacteria cause superbugs because of phage therapy to one bacteria? Will it cause superbugs to other bacteria?
That is a little bit less likely, in my opinion, because these interactions are also fairly specific. There are phages which are bacteria specific. So, for example, there's a microbacteriophage. This microbacteriophage will not affect other bacteria. So, it will target something that is specific in mycobacteria. Most of the antibiotics we have are not that specific. They will kill all bacteria. Therefore, resistance mechanisms can also be fairly widespread across the bacterial kingdom. In phages, I don't know the exact answer to your question, but my guess is that it will be less prevalent.
Okay, sir. Thank you. Another question. I heard that there will be some side effects due to the use of antibiotics. To combat the side effects, they will give the medical practitioners some other drugs to combat that. So, will this also leads to development of superbugs?
Not necessarily. See, antibiotics will have side effects for other reasons, because our body is not tuned to receive these drugs, right? And we will react to the drug. Now the side effects, the other medicine will be given for the side effect itself. So if you're vomiting, for example, your body recognizes that the antibiotic is foreign, and a normal response is to induce vomit, for example. Now, if you're also given a drug that is that prevents you from vomiting, that need not directly lead to result in antibiotic resistance.
Sir, one more last question. On the external part of the human beings, some infections which are caused by some fungi and algae, the doctors prescribe that don't use soap and other thing, and sometimes they prescribe antibiotics also. What will be your view on not using soaps and using some antibiotics specifically to treat the external infections?
I don't know. I need to know the specific context in which the doctor made his recommendation. I don't know enough to. I don't think we can generalize it like that. It must be a specific context in which this advice was given.
Okay, sir.
But in general, I mean, this is a very personal opinion. In general, this, a very bland kind of soap will be better than something that has too many synthetic flavors and chemicals.
Okay, sir. Thank you. Thank you for your very informative and enlightening and interactive talk. Finally, I would like to thank today's speaker, Dr. Varajan Sundarmurthy, for sparing your valuable time and highlighting on antibiotic misuse and rise of antibiotics, rise of superbugs, in spite of your busy schedule. Thank you very much.
Oh, thank you very much. It was a pleasure.
Thank you one and all for joining to this program. As we have come to end of program, thank you one and all. Thank you.
Bye.